Okay. So good morning, everyone. Um, I wanted to start by saying a big thank you to Alex for the invitation to speak today and for organizing this really exciting meeting. So my name is Seham Ebrahim. I'm an assistant professor at the University of Virginia. I started my lab in 2022, so it's been 3 and a bit years. And today I'm going to share with you some work that we have been doing on mechanosensitive motors and talk about how spatial organization of these mechanosensitive signaling is important across tissues. So, yeah, so I'm going to start by really just giving you a very brief introduction to our lab, the type of research we're interested in, and our approach. And then I will talk to you about a story. This is all new, unpublished data on how spatially encoded mechanical signaling determines cell function and development. So to start with, our lab is really interested in this idea of forces. So if you think about any given cell or tissue in your body at any given time, you can imagine that it's experiencing some type of force, whether it's your heart beating, your bones growing, or your gut digesting the treats that you just ate. And so we're becoming increasingly interested in this question of how cells are able to generate, sense, and transmit these forces, and how that impacts their decision making or their physiological function, and how things go awry in the context of disease. And because this is a very broad, overarching question, we are interested specifically in the role of the cytoskeleton, specifically the actomyosin cytoskeleton, and this family of proteins called septins, as well as a family of putative mechanosensory membrane proteins called TMCs. And that's what I'll be talking to you about today. So when I talk about spatial biology today, it's more in the context of subcellular spatial organization. And so we look at our questions across scales. I like to call it cellular physiology across scales. And so a lot of work that people have done has been in the context of isolated cells in a petri dish. And you can imagine that these type of studies have been instrumental in gaining mechanistic insights into several cellular functions. However, if you look at the same cells that are in a petri dish and compare those to cells that are in the context of intact tissue within the body, you can see that they're completely different environments and ecosystems. So epithelial cells growing isolated in a dish, even if it's a co-culture, are never going to experience the same environment as epithelial cells that are lining your GI tract, which are experiencing forces from peristalsis, crosstalk with the microbiome, information exchange, hormone regulation, etc. And so what we in my lab do is we try to look at the questions that we're interested in vivo in the context of intact cells and tissues, in organoids, and also in isolated cells and culture to gain kind of a very holistic picture of our question. And the approaches that we use across scales include super resolution and live imaging, machine learning and artificial-based, intelligence-based image analysis, classical biochemical approaches, as well as omics approaches. And so why spatial context, even in a subcellular level, why does it matter? So I like to give this simple example. So here in magenta, you can see a cell that's expressing F-actin, or rather F-actin in that cell is labeled with phalloidin in magenta. And you can see that this F-actin forms these two-dimensional networks that have been studied for several decades and that I think most of you are familiar with. So these can include sort of lattice-like organization at the leading edge of the cell, actin philopodia at bundled actin that form philopodia. But now if you look at the same F-actin in magenta, but in the context of different cells in intact tissues in the body, you will see that they form strikingly distinct architectures. And these architectures are formed because the proteins, it's the same protein, but in these different spatial contexts have to perform different functions. And these are images that were taken throughout the course of my training. So the first one is just showing you exocytic vesicles in the pancreas, and you can see that the actin and magenta forms this almost soccer ball-like lattice. It's very different from any kind of actin organization that you would see in the cell in culture. Then you can see these are stereocilia, which are actin-based protrusions that are actually mechanosensory, and I'll talk about this a little bit later in the talk. And then finally, you can see that actin and myosin in epithelial cells form these kind of mini sarcomeres that connect adjacent cells, and the expansion and contraction of these sarcomeres affect epithelial cell morphology and function. So this just hit home to me the importance of studying the spatial context of proteins within cells. And we also do that using live imaging approaches. And so here are just a few examples of high-resolution intravital microscopy from the lab, again, across different organ systems. And in each case, you can see almost the same protein that is labeled, but you can see how it localizes and functions differently depending on the spatial context. So that's just a brief introduction to the interests of the lab and the approaches that we use. So today I'm going to tell you specifically about one story that I think is particularly interesting in the lab, and that's this question of how spatially encoded mechanical signaling determines cell function and development. So specifically, I'm going to talk to you about this family of proteins called TMCs. And so this image is from a relatively recent review on mechanosensitive transmembrane proteins. And you can see TMC is highlighted there in the red dotted box. So TMC, there are 8 members of this family. They stand for transmembrane channel-like family. They have 10 transmembrane domains, and they're expressed in protozoa all the way up to animals. Now, of these 8 members, by far the best studied is this protein called TMC1. It's sort of the canonical member of the family. And so TMC1 is basically believed to be the pore of a mechanotransduction complex that resides at the tips of those stereocilia that I showed you in a previous slide. So just an introduction to what stereocilia are. Stereocilia are these actin-based protrusions. They have this remarkable staircase-like organization, and they sit on top of sensory hair cells of your inner ear, right there. And so these cells basically convert mechanical signals such as sound or head motion into electrical signals that can be understood by the brain. So how does that happen? So at the tips of each of these stereocilia sits a channel, mechanosensitive channel. And what happens is when the stereocilia bundle is moved in response to mechanical stimulation, this channel opens and calcium ions flow into the cell, and the cell is depolarized and signals are sent to the brain. And so TMC1 is thought to be the pore, the calcium-sensitive selective pore of this mechanotransduction channel. And in this movie, I'm showing you 2 stereocilia bundles, and you can see that these cells express GCAMP3, which is a marker for calcium. And this is just to show you spontaneous calcium entry into the stereocilia by way of this TMC-mediated mechanotransduction channel. So during my training, I discovered that TMC1 is localized at the tips of the stereocilia where the mechanotransduction channel was thought to be located. So all of this information about TMC1 that is known has been based largely on genetic, physiological, and some computational studies. The main bottleneck in the field remains that there's currently no direct evidence for the structure or the molecular function of mammalian TNCs. And the reason for this is, or the main 2 reasons for this are, firstly, these hair cells, these sensory cells of your inner ear, they're very, very few in number. So in each ear, you have in the range of 15 to 20,000 cells only. That's not enough to purify the complex and study its physical properties. Additionally, trying to overexpress TMCs in a heterologous system such as HEC cells has proved unsuccessful. So here's just an example showing that when attempts have been made to express TMCs in cell lines, the TMC do not traffic to the plasma membrane where they would need to be in order to function, but instead they get retained in the ER. And so this remains a big bottleneck in the field because people still cannot definitively say that TMCs are indeed mechanosensitive calcium ion channels. And so this was one of the questions that intrigued us in our lab. And so we were inspired by this particular paper that came out a couple of years ago, where this group took an alternative strategy to understand the biophysical properties of TMCs. And so what they did here is they turned to a model organism, C. elegans, which expresses a homologue of TMC1, which is C. elegans TMC1, which is a homologue of mammalian TMC3. And so what they did here is they essentially tagged a GFP to the endogenous TMC1 and were able to grow up a lot of worms, 80 grams of worms in this case, and they were able to purify this native TMC complex. And so what you can see here, that's predicted that the TMC1 forms a homodimer. And importantly, it does not act alone. It has these accessory proteins, TIMI, as well as this protein called COM1, which is mammalian CYP1, which will become important a little bit later in the talk. And important to know that mutations in any of these proteins, TMC1, TIMI, or COM1, CYP1, cause deafness and vestibular disorders in humans. And so we wanted to take a slightly different approach. We wanted to see if we could learn about other TMC family proteins that are expressed in more abundant or more easily accessible tissues or cells. And so one TMC that caught our eye was TMC5, which is highlighted here. And we found that it was highly expressed in enterocytes, intestinal epithelial cells, as well as spermatids. And today I'm going to tell you about the work that we've been doing on TMC5 in the context of spermatids. So the first thing we did was to validate this data, which we got from the Human Protein Atlas. And so in collaboration with Brian Herman at the University of Texas, we confirmed that TMC5 in the testes is exclusively expressed in spermatids, in both mouse as well as human tissue. And so I'm not sure how good your germ cell biology is. So what are spermatids? Just a very, very brief introduction. So if you look at the seminiferous tubules in the testes, in any tubule, you will see germ cells at different stages of development. And so here's a schematic showing just that. And so you basically have spermatogonia up here, then they differentiate into spermatocytes, and eventually you end up with this haploid round spermatid stage shown here in blue. And then these spermatids, the round spermatids, undergo this very striking morphological change to become the elongated spermatids that I think everybody is familiar with. And so here's just an example, of what that looks like by confocal microscopy. So you can see in the low mag image, tubules containing germ cells at different stages of development. And in the high mag image, you can see the round spermatids here based on their nuclei in blue, and then the elongated spermatids where you can see the nuclei are now very condensed. You have this mid piece that is decorated with mitochondria and this beautifully long tails. And so this stage is where the TMC5 is expressed exclusively. And so if you look a little bit more closely at this stage, you can see that in order for that round spermatid to elongate into the developed spermatid is a highly force-dependent process. And so we looked in the literature to see if these round spermatids actually respond to mechanical forces at all, and there was nothing in the literature. And so the first thing that we wanted to test was that question: do spermatids respond to mechanical stimulation? And the approach that we decided to take was to use high osmomechanical stimulation. So basically putting the cell in a hypotonic solution. And so what that does is cause the cell to swell and causes the membrane to stretch, as you can see in this schematic here. And so what we hypothesized would happen is, should the cell respond to mechanical stretch, that a channel would open, allowing calcium to flow into the cell. And so to test this, we used a calcium dye called Fura-2, and we imaged isolated spermatids. I'm showing you here an example. And so any unbound calcium is red in these movies, and calcium bound is green. And so here you can see two spermatids that look yellow because they have a little bit of bound and a little bit of unbound calcium. And so what we do is then we stimulate with this hypoosmotic buffer. And what I hope you can see is at the point where we do the stimulation, the cells turn green, suggesting that there is indeed a calcium influx in response to this osmotic stimulation. And that's quantified here. So we were very excited by this result because this is actually the first time somebody has shown that spermatids can respond to mechanical stimulation. And you can see in the bottom graph there that when cells are put in buffer that does not contain calcium, you do not see that calcium spike, suggesting that that calcium influx is from external calcium. And so the obvious question was, well, could it be due to the presence of a different mechanosensitive channel? And so we looked at all known mechanosensitive channels, including piezos, TRPC, TRPM, and TRPV channels. And there was no known mechanosensitive channel that are expressed at that spermatid stage. And so this led us to ask the question, so is this response of spermatids to membrane stretch forces mediated by TMC1? And so to address that question, we developed a couple of mouse models. So we have TMC5 M-cherry mice. So these are mice with an M-cherry reporter tagged to the endogenous TMC5, as well as a TMC5 knockout animal. And so the first thing we wanted to do was to evaluate where TMC5 is localized in the spermatids and if that corresponds to the RNA, single-cell RNA-seq data. And so looking at testes cross-sections from the mice expressing TMC5 M-cherry, you can clearly see that TMC5 M-cherry is expressed at the plasma membrane of these round and elongated spermatids. You can see the signal in red. And so that confirms that is indeed a plasma membrane protein. The next question we had, well, so TMC1 is thought to be mechanosensitive and it forms this complex. Does TMC5 form a similar complex in spermatids? There's no structure for either of these proteins. So based on homology modeling, we first determined that TMC5 is predicted to be very similar to TMC1, especially in the putative pore domain. And so this was not an unreasonable hypothesis. And to test the hypothesis, what we did was we used our TMC5 M-cherry mouse. We basically dissected out the testes from this mouse and used that tissue to purify the TMC5 M-cherry complex by binding it to beads that had antibodies against M-cherry. We then used mass spectrometry to identify the TMC5 interactome and then validated with IF. And so what we found very intriguingly was that TMC5 interacts based on this experiment with SIB proteins very similar to the TMC1 mechanotransduction complex in the inner ear, as you can see from this table. We then looked at where SIBs are expressed in the testes, and we found that they are also expressed on the plasma membrane, similar to TMC5. And strikingly, when you knock out TMC5, that membrane localization of TMC1 is a TMC5 of SIB1 is also lost, validating that these two proteins interact in vivo. And here is biochemical validation that when you pull down TMC5 M-cherry from the testis, that SIB1 co-IPs with it. So this is all very interesting. And also, AlphaFold predicts that these four proteins interact forming a heterodimer. So TMC5 CYP1, TMC7, and CYP4 all form a heterodimer, which we thought was interesting, and became even more interesting when we discovered that mutations in any one of these proteins, CYP1, CYP4, or TMC7, were all linked to male infertility. That kind of supports the idea of this heterocomplex formation. And so this led us to the question, well, what about TMC5? And indeed, the TMC5 knockout mouse is also infertile. So why is it infertile? So if we go back to that testis that I showed you, where you have beautiful round spermatids and elongated spermatids in the wild type, looking at the TMC5 knockout testis, we see that that spermatid elongation process, that force-dependent elongation of round spermatids to elongated spermatids, is disrupted in the TMC5 knockout. And then the obvious question was, well, is this because of a lack of mechanical response in these round spermatids? And I'll just skip this just to get to that. And so we basically repeated the calcium imaging in hypotonic buffer experiment once again. And just to remind you that in the wild type case, you see this calcium spike in response to the mechanical stimulation. However, in the TMC5 knockout, that calcium response is completely abrogated. And so this suggests to us, the summary so far is that we discovered that developing spermatids actively respond to mechanical perturbation, that mechanosensitive signaling originates at the plasma membrane, that TMC5 is spatially restricted to the plasma membrane, and that loss of this TMC5 abolishes the mechanically evoked calcium influx, arrests spermatid elongation, and results in male infertility. So these are all very exciting results, but of course, it leads to the question about how this is all working. And so what we wanted to do was to test how this membrane-initiated calcium signal potentially shapes spermatid transcriptional identity. And to address this question, we turned to single-cell RNA-seq. And here you can see from this map already that there's a population of cells that are depleted or highly significantly reduced in the TMC5 knockout, which, oops, you can see I'm close to the end, which is what you would expect to see based on our imaging data. So we're losing a lot of the late round spermatids. So just to quantify that here, you can see that this population of cells, which makes up about 50% of the total cells in the testes, is reduced to about 10%. So looking at the differentially expressed genes in the spermatids that are left over, we also found that there was a number of genes that were significantly downregulated. And a collaborator of ours pointed out that several of these genes are targets of CREM-tau, which is a master regulator specific to spermiogenesis and whose activity is dependent on calcium signaling. So we were interested to see if there was that was the underlying cause of this arrest of spermiogenesis. And so we did some differential gene set enrichment analysis, and basically we found that that was indeed the case. The genes that are downregulated in the CREM-tau knockout testes are also downregulated in the TMC5 knockout testes. And so the conclusion there was that TMC5 dependent calcium signaling is required to support CREM-tau associated transcriptional programs during postmeiotic differentiation. And so the take home from this was that force-dependent calcium signaling on the spermatid plasma membrane is linked to transcriptional control. And these findings extend the functional scope of TMC-based signaling from sensory epithelia to germ cells, showing that the same family of proteins can have completely different functions and physiological consequences in different cell types. And so with that, I would just like to thank you all for your attention, to thank my lab, specifically Sharon Zhang, who's a graduate student in my lab who led this project, and our funding sources. Thank you.
Okay, now you can hear me. Thank you. Beautiful talk. I'm wondering that actually I've been wondering for a while why TMC7 why they're not proven yet, but you give the reason. So about the TMC5, are they the only or the major calcium channel available in the spermatide?
That's a great question. So as far as we know, there is no evidence, compelling evidence in the literature for spermatids being sensitive or being able to transduce calcium. So yeah, as far as we know, although we haven't proven that it is indeed a calcium ion channel, it is, as far as we know, the only protein that has been identified to regulate calcium so far. That's not true for mature sperm, where you have channels that like study in the case of these developing sperm.
The questions?
Oh, thank you. Yeah, just seconding beautiful images, beautiful microscopy, and really exciting science. So kind of taking a little bit step back, but you showed a lot of different technologies and modalities. Where do you see bringing some of the spatial omics into the dynamic imaging and multiphoton? Like where could that fit and how might you integrate those data sets? If you could speak just globally, broadly about data
Yeah.
integration,
I mean,
different modalities.
That's a great question. And that's one of the reasons I was very excited also to attend the seminar and to get feedback from the audience about where I think there is scope to incorporate those approaches, especially in the context of germ cell biology. I feel like there's not much out there yet, but it's definitely an area I'd be interested to explore.